Hello friends, welcome you in my YouTube channel. I am Dr. Mohammad Sirajul Faizi, Department of Chemistry, LS College Muzaffarpur. Today in this video, I am going to explain about medicinal chemistry. So, I will talk on organic chemistry, isomers, functional groups, steroids, and the drug discovery concept, general strategy. In 4th century, Hippocrates boiled willow bark to make tea. This found that it helped people with fevers. Later, scientists extracted yellow crystals from willow bark and called it salicine after Salix alba. Genus species of willow found that this compound reduced fever and inflammation, side effects, stomach irritations, and bad taste. So, name this compound. Okay. So, that compound is aspirin or 2-acetyloxybenzoic acid or acetyl salicylic acid. The structural formula of aspirin, acetyl salicylic acid or ASA is this. This is ester linkage, this is acid. Now, organic chemistry, the largest subdiscipline of chemistry, is organic chemistry, devoted to the study of carbon based compounds. How many compounds known? 40 million known compounds, and 12 million of them are organic. 40 millions known organic compounds and 12 million of them are organic. You see how much organic compound. So look at here: caffeine, Aleve, Viagra, Word, LSD, tryptophan, THC, aspartame. Isomers. Isomers have the same chemical formula but different molecular structure and properties. This is N-butane, this is the T-butane. So how many isomers does C5H12 have? Draw them. Now functional groups impart characteristic physical and chemical properties. In all drugs, it is these functional groups which gives the chemical its physiological activity. Alcohol, aldehyde, ethers, ketone, carboxylic acid, and so on. You see, alcohol, aldehyde, ketones, carboxylic acid, ester, amide, etc. So look at aspirin. Aspirin contains benzene ring, carboxylic acid, ester. This is ester, carboxylic acid, and benzene. Ester, carboxylic acid, and benzene. This is a structure of aspirin. One allows the molecules to get into your system. This is, and 2 plus 3 are active drug parts. This is the active drug parts. So aspirin contains this and another medicine. Now aspirin, how it works? Body, 2 types of communication system. First, nerves, breathing, reflux, heartbeat. Second, chemicals, hormones, release chemicals into bloodstream, epinephrine, insulin. Aspirin blocks cyclooxygenase enzymes from producing prostaglandins. Prostaglandins produce fever and swelling, increase sensitivity to pain inhibit vessel dilations. You see, aspirin blocks cyclooxygenase enzyme from producing prostaglandins, and prostaglandins produce fever and swelling, increase sensitivity to pain, inhibit vessels dilation. This is membrane bound receptor target cell. Hormone molecules, capillary blood, plasma membrane. Drug design. Goal: to design, engineer a compound so that its beneficial effects are enhanced while the side effects are decreased. Willow-Barty aspirin. Relate chemical structure to drug activity. Two groups of drug. One, those that produce a physiological response, aspirin hormones, psychoactive drugs. Second, inhibit the growth of substances that cause infections, antibiotics. Now, drug. Example, morphine, highly additive. Demerol, same active areas as morphine, not as additive. You see the morphine active area. So drug or molecules, cell membrane and receptor site. This is drug or hormones and cell membrane receptor site. So drug steroids. All steroids have the same backbone structure. You see the 6 member, 6 member, 6 member, 5 member. All steroids have the same skeleton. 1930, in order to study testosterone, they had to process 1 ton of bull testicles to yield 5 milligrams, 4 ton of pig ovaries to yield 12 grams stone. So, birth control. You see progesterone and norethindrel. Only difference is here functional groups. So as with aspirin, birth control drugs came about through the molecular modifications. So drug discovery concept, general strategy. You see the drug discovery process, target identification, 3 months to 2 years hits, 3 to 4 months active to hit, 3 months hit to lead, and 6 to 9 months new lead optimizations project, and 2 years candidate drug CD. So these are steps for the discovery of drug. So lead compound from a variety of sources, chance discovery, penicillin, natural products, clinical observations, natural ligands, existing drugs, and high throughput screening. You see the Taxol, Viagra, penicillin, all organic compounds. So natural ligands are adrenaline, if methyl then non-adrenaline. So formoterol, AstraZeneca, catechol, isosters, this is toxicity, and now this is your catechol, bioisoster, toxicity, increased size, selectivity, and duration. Now existing drug, also known as me too or me better approach. You see Pfizer making this Viagra, short duration, multiple side effect, and incompatibility with other drugs. Beware patent issues. This is Bayer and Cialis, Eli Lilly. So fewer side effects and incompatibility with other drugs. 30 hours duration, the weekend pill. High throughput screening, STS, an industrialized process which brings together validated tractable targets and chemical diversity to rapidly identify novel lead compounds for early phase drug discovery. 50 to 60% of new drugs projects originate from ASTS. How? Validate tractable targets. Target selected for the STS, industrialized process, STS, assay technology and automations, and chemical diversity, sample selection for STS. To establishing a STS, you have the human and pathogen genomes, target ID, validated tractable targets. Then ST screen developments, microliter plates, STS test tubes, you see this way, 96 well, 300 to 100 microliter, 9 pitch, 384 well, 25 microliter, 2.5 pitch, and this is the, and same way high throughput radiations banding assay, SPA scintillations proximity assay, you see high throughput fluorimeters, how it work, establishing ASTS, library chemistry, three most commonly used reactions, amide coupling, reductive aminations, sulfonamide formations. So amide coupling reactions, sulfonamide formations, and reductive aminations. So in this video I already discussed organic chemistry, isomer, functional group, steroid and drug discovery concepts. Hope this video will be helpful to you. If you like this video, then subscribe my channel, and thanks for watching.